So this is dihybrid inheritance. In a previous presentation, we went through monohybrid inheritance. Monohybrid, of course, is one gene. Dihybrid will be two genes. So on the previous monohybrid presentation, we went through a number of terminologies that you really must memorize before you can progress on to any of the exam questions on inheritance. This is the same list again. Obviously, this time the definitions are covered up. Take a few moments to go through each of those words, what you think the definition is, and then we'll progress on and make sure that you've actually memorized them correctly. If you do get any wrong, folks, I'd strongly recommend you keep going over this slide until you get 100% right, because you're going to find it very difficult to progress through if you haven't memorized these words. So by now you should hopefully have taken a little time, pause the video, and gone through each of the different words. So just as a quick review, here are the definitions for each of the terminologies that you must memorize. You're going to find it very difficult to go through and answer any of the questions if you haven't gone through these words in advance. So a simple question, hopefully most of you would be familiar with this face from GCSE biology, but a simple question is, who is the person pictured and what is their connection to patterns of inheritance? The picture, of course, is Gregor Mendel. If you're not familiar with Gregor Mendel, I strongly recommend you do some background reading on the individual. But his most famous piece of work is with pea plants and the inheritance patterns in pea plants. Most recently, thanks to statistical analysis, it's actually believed that his research may be a little too perfect. The accusation isn't that he made his results up or anything like that, more a case of that the ability to distinguish between what a tall plant was and what a small plant was may have been a little bit more difficult, and he may have tended to have a little bias when giving his results. But again, folks, please do research that if it does pique your interest. Just to go back over this simple monohybrid inheritance, in front of you what is a simple diagram showing two different phenotypes, the tall and short plant. Each of those phenotypes is controlled by one gene, which is in this case being given the letter T. The capital T, of course, is the dominant, the lowercase t is the recessive. What has happened is you can clearly see that this characteristic has one tall plant, which is homozygous dominant, one short plant, which is homozygous recessive. When those two plants are bred together, they produce an entire generation of heterozygous plants. In the bottom half of the screen, you can see a Punnett Square, which shows you each of the different alleles coming together from each parent to form the genotypes of each individual organism. And you can see that no matter which alleles come together, that the offspring will all have the same genotype and hence will all have the same phenotype. In this particular section, what we've done is we've taken the phenotype that was bred in the previous slide, and we've taken two of those heterozygous parents and we've bred them together. In this particular instance, the two heterozygous parents produces a 75% tall population and a 25% short population. Once again, if you look at the bottom half of the Punnett Square, you can see how the alleles combine together to give you a homozygous dominant, two heterozygous conditions, and one homozygous recessive. However, in terms of phenotype, the homozygous dominant and the heterozygous conditions will give you the same phenotype, which is tall, compared to the two little t's, which gives you the recessive phenotype, the homozygous recessive, and hence we end up with 25% of the population, approximately, which will be short plants. Just to give you a brief idea of the kind of basic differences between monohybrid and dihybrid. As we said previously, monohybrid is one gene where dihybrid involves two. In monohybrid, we have only one characteristic because we're only looking at one gene, whereas in dihybrid, we're actually looking at two different characteristics. And it's extremely important that we recognize that those characteristics do not occur on the same chromosome. And we will go into that pattern of inheritance at a later time. In monohybrid inheritance, we have two possible gametes, whereas in the dihybrid inheritance, we always have four possible gametes. Again, they can be the same in terms of genotype, but there's always four possibilities with dihybrid, where in monohybrid, there's only two possible gametes. With the dihybrid, again, we'll have two polypeptides because we're dealing with two genes, And in the dihybrid or Punnett Square will be a 5x5 grid compared to the 3x3 grid needed for the parents and different combinations of offspring in monohybrid inheritance. So on this slide, what we've got is the dihybrid inheritance pattern seen with two specific genes. One of the genes controls if the seeds are green or yellow, and the other one controls if the seeds are smooth or wrinkled. First thing we have to do is we have to allocate them a specific letter, and that letter needs to be something that's easily identifiable when it's in upper and lower case. So you need to be careful which letter you pick. Things like W's are not very good. Things like Y's and S's are not very good. So you're going to look for two letters, one for to control the color, one for smooth and wrinkled, and you need to determine the dominant and recessive characteristic. So if you look through the text says, carrying a cross between two parents. One is homozygous dominant for both conditions, and one who's homozygous recessive for both conditions. So we know straight off the genotype of the two parents. It also tells us that yellow seeds and smooth seeds are dominant characteristic. So our smooth seeds here and our yellow seeds here both need to be capital letters. It doesn't matter which letter we pick. We haven't been given one on the question, but we have to make sure that both the smooth and the yellow are capital letters with green and wrinkled lowercase. On this slide, what I've done is I've given you the kind of format that I would recommend you use when answering inheritance style questions on the exam. First thing you do is you identify the letters represented each of the alleles for each of the different genes. In the question, it says there are two different genes relating to two different characteristics. One is color and one is smooth and wrinkled. And what I've done is allocated a capital and lowercase g for the color and a large and lowercase b for the smooth or wrinkled. Again, the reason we didn't pick S or W or Y is because the uppercase and lowercase letters can look the same. So once we've identified the different alleles for each of the genes, we're then going to put together our parental genotypes. One of the parents is in the question identified as being homozygous for both conditions. So therefore, we simply give it two capital G's and two capital B's, remembering that each gene has two alleles for each organism. In the other adult, we have the homozygous recessive for both conditions, and again, we simply give two alleles for each of the different characteristics, so two lowercase g's and two lowercase b's. When it comes to putting the gametes together, remember dihybrid, there are 4 possible gametes for every single organism. However, you will find that sometimes the gametes will be exactly the same in terms of the genotype. So what I would recommend is you always set out all of the gametes until you can kind of wrap your brain around the questions, and then you can shortcut. But for the first parent, all of the gametes will be uppercase G and an uppercase B, whereas in the other parent, all of the alleles will be lowercase for each one. When we put that arrangement into a Punnett square with one parent along the top, one parent down the side, we look at that arrangement, and then when we do all our crosses, all of the individual organisms will be heterozygous for both conditions. And if we look back at the top, we can say that all of the offspring will be yellow and all of the offspring will also be smooth. Ideally, I would like to have the bottom phenotype box on the same slide as the previous slide, but it just wouldn't fit, so I've set them out for no other reason than so they're readable. But in your exam question, this last bit called phenotype should be included in your answer just as a continuation of the first part. And what you simply identify is all the possible phenotypes that could be combined, which is the right column. And then what you do is you simply identify each of the different genotypes that exist inside your Punnett square and the corresponding phenotypes. As all of our organisms have exactly the same genotype, they will all be yellow and smooth. In the exam, what is most common after you've done the first cross is that they ask you to take 2 of the offspring and to do another breed with 2 of the offspring. In the previous question, the offspring were both heterozygous, and they were heterozygous for both conditions. Now I've put one of the parents in blue on the screen. In the exam you wouldn't be expected to do that. That's purely because I can do it in this presentation. In your exam, if you did everything in black ink as you're meant to write in black ink, that's absolutely fine. As long as you've got the right letters in the right place and everything labeled, there should be no issues. In terms of the parent 1, there are 4 possible combinations. Each of those letter G's can independently join with each of those letter B's. There's no preference, there's no predetermined association, they're not linked in any way, they're all completely independent. So we can have any possible combination, but we always have to have one letter G in a gamete and one letter B in a gamete. As a consequence, we have 4 possible outcomes. We've got the 2 capital letters, a recessive for one characteristic and a dominant for the other. A dominant and a recessive are 2 recessives. As parent 2 is also heterozygous, you've got exactly the same gamete combinations. Just as before, we put those parents into the first row and column of a Punnett square, and we end up with all the following possible combinations. Now, when writing in combinations, I would always suggest that you do a row or a column at a time. If you try doing each box individually, it's very easy to make simple mistakes, which can cause all sorts of problems in subsequent parts of the exam question. So I would always do a complete column or a complete row and write in the same letter all the way across, so you know that you're not adding in or missing out on any of the alleles. And you can see that each of the offspring has 2 letter G's and 2 letter B's, and each of the offspring has inherited 2 alleles from parent 1 and 2 alleles from parent 2. What we then move on to is we then start allocating each of the offspring genotypes and the respective phenotypes. Now that I've identified all of the genotypes, I need to make sure that I match them up to the relevant phenotypes. The method I would suggest using is literally going through each of the individual sections of the Punnett square and allocating each one of the genotypes to the correct row in a list of the different phenotypes. Once you've done that, you'll know you've got it right if you have 2 heterozygous parents and the parents are both heterozygous for both conditions. You will get what's called a standard 9:3:3:1 ratio. 9 of the outcomes should be dominant for both characteristics. 3 outcomes will be dominant for one characteristic and recessive for the other. Another 3 outcomes will be recessive and then dominant, whereas 1 of the outcomes will be recessive for both characteristics. So previously I mentioned the fact that each of the letter G's and B's could be independently passed on into the gametes. They were not linked in any way. What that means is we have separate chromosomes for each allele, but we also have separate chromosomes for each of the genes. That means that whatever letter G is passed on to the offspring does not in any way impact on the letter B that is passed on. So they're completely separate characteristics or they're unlinked characteristics. And what we refer to there is what's called the law of independent assortment. And it's that each of the pairs of alleles will combine independently of each other inside a gamete. Another type of question, or possibly a follow-up to the previous inheritance type question that could appear in the exam, is where you're asked to work out the number of organisms that would have each of the possible phenotype combinations. Again, this will work if you have the ratios. I've gone with the standard 9:3:3:1 ratio, which is 2 heterozygous parents, and both parents are heterozygous for both genes. If that is the case, we have 16 possible outcomes, and we simply work out that if we've 1,600 organisms, each one in the ratio, it represents 100 organisms. When we do that, we will find that we're able to work out the individual numbers of the organisms that we would expect to find, because of course in real life you will not find exactly 900, exactly 300, exactly 300, and exactly 100 of each. And that leads into the whole concept of statistical testing, which is covered in another presentation. So as I mentioned previously, we will not get exactly the ratio numbers that we're expecting if we had a population of 600. It'd be slightly more, slightly less of each of the phenotypes. And the pure reason for that is simply random chance. We have 4 possible gametes, but each of those 4 gametes is equally likely to successfully fertilize, and sometimes by pure random chance, you happen to get incidences of where things occur over and over and over again. So you can end up with families with 5 or 6 girls in them, but yet somewhere else in the world you'll also have a family with 5 or 6 boys in it. It was pure random chance. But when you get large enough numbers, you should find that your ratios should match up with the population pretty accurately. And again, that's where the statistical testing comes into play. That's the end of the dihybrid presentation.